We'll now look at some of the basic mechanisms of neuronal communication. We'll delve into more detail about how different neurotransmitters affect neuronal communication in the next chapter. This gives you the overall schematic of the neuron in a very simplified sort of way. We envision multiple inputs synapsing on the dendrites. Ultimately, the neuron produces an output through its axon to continue this process at the next synapse. The process of transcription and translation from the nucleus to the cytoplasm is shown to remind us that the end result of many of these aspects of neuronal communication is to change the gene expression within the neuron. So in the lower part of the figure in the book, we see the three major systems that we'll be dealing with in this course. Number one shows an ionotropic channel. By that, I mean when the neurotransmitter, the black dot, attaches to the receptor, it causes a conformational change such that a channel opens. Ions then flow back and forth through this channel, and that usually has some very direct input on the firing rate of the neuron, either to trigger a action potential or to cause hyperpolarization and prevent the neuron from firing. In contrast, the G protein second messenger system works through proteins that ultimately will change the long-term permeability of ion channels or will increase or decrease transcription and translation of various genes. Now, a system that you're probably less familiar with is the tyrosine kinase linked receptor system, or TRK system. Now, these have different types of a different type of neurotransmitter from the G protein system. The G protein system are the small molecule neurotransmitters that you're very familiar with. In contrast, the growth factors tend to be very small polypeptide chains. Although they're obviously larger than a neurotransmitter, they're still much smaller than many of the formal hormones you're used to dealing with. The growth factors generally will operate at a greater distance than do the neurotransmitters, which tend to be confined to the region around their receptors. When two growth factors attach to the TRK receptor, we again get activation of or repression of gene expression. And then finally, we have the various steroid hormones that do not need a receptor at the cell membrane. Indeed, they diffuse directly through the cell membrane and attach to a steroid receptor in the cytoplasm. This complex then generally enters the nucleus, where again you get influence of gene transcriptions. Cortisol is perhaps the best known of these steroid neurotransmitters, and we'll study that in a great deal when we look at the stress response. But there are a number of other so-called neurosteroids that also work through this system that may be highly relevant to psychiatric illness. The next thing to bear in mind is the other cells that attend the neuron. These include the microglia that really act as scavengers that also may release cytokines, and they are part of the inflammation process. The oligodendrocyte, which is responsible for the myelin, and perhaps most important for our purpose, the astrocyte. Now, these were often thought of as simply neurons, neuroglial cells that simply nurtured the neuron. So they would often have one astrocyte foot on the blood vessel, the other would be around the synapse. But it's now very clear that the astrocytes are involved directly in neuronal communication. We'll go into a little more detail here than what is in the book. I've mentioned already that the astrocytes simply provide extracellular support to the neurons themselves. They participate with immune modulation responses with microglia. We'll get into that with affective disorders and PTSD in the future. They regulate the blood-brain barrier by virtue of these feet that they have on the blood vessels themselves. They're involved in the neuron's energy metabolism, as well as regulating the amount of potassium and sodium. They're involved in the reuptake of neurotransmitters. In fact, we'll see this very clearly with glutamate, where astrocytes play perhaps the predominant role in reuptake of that neurotransmitter, but it turns out this also is important for serotonin and norepinephrine. One that is not quite mentioned here is these calcium waves that involve a parallel method of neuronal communication. So here we come back to the book to figure 3.2. You can see here that a neuron, the presynaptic neuron in number 1, releases a neurotransmitter. That neurotransmitter attaches to the postsynaptic receptor in the postsynaptic neuron, but also to a postsynaptic receptor on the glial cell. Through a variety of second messenger systems, the astrocyte can create a calcium wave that allows it to communicate with other astrocytes throughout the brain. And you can see this very clearly here in this small video. The red represents the calcium, and you will see how precise the projection of these calcium waves are from second to second. Notice how sometimes they're down only one particular branch of the astrocyte. And the calcium waves vary in their density and their length, as well as the time. There you see that one just shot out and came back again. So in addition to that, the astrocytes will release so-called glial transmitters into the synapse that attach to their own particular receptors on the neurons. And this makes the synaptic processes much more complex than we ever realized. And astrocyte pathology is thought to be involved in a wide range of neuropsychiatric diseases. Now let's review some of our basic neurophysiology. The upper left in A, let's imagine a cell with potassium chloride and sodium channels, so those ions can move in and out. Let's put 2 millimolar of potassium chloride in the cell. This will just redistribute itself inside and outside the cell. Now in B, let's add 120 millimolar of potassium that is bound to a negatively charged ionic protein. Now this protein can't just redistribute itself, it's stuck inside the cell, so a small amount of potassium will leak out, but then it is opposed by the electrostatic gradient that is building up. So you end up with a potential across the membrane of negative 85 millivolts, and this is shown in C, but that situation can't go on for very long because the cell would burst. There's too much osmotic pressure inside the cell. So in D, we add 130 millimolar of sodium chloride outside the cell, and both the sodium and chloride will distribute itself such that we reach an equilibrium with the typical neuronal cell membrane potential of negative 70 millivolts. Now the cell will eventually leak, So this membrane potential is maintained by the active sodium and potassium pump. Now let's take a look at the basic principles behind the action potential. In A, when the neuron's at rest, we have the passive channels. Those are open there. Those are the ones we just spoke of. Uh, to this mix, we'll now add the voltage-gated potassium and sodium channels. These are currently closed. And then we have the ligand-gated ion channel. This will only open when the neurotransmitter attaches to it. You can see that we have the ionic negatively charged proteins on the inside, potassium on the inside, and more sodium on the outside. So now the neurotransmitter binds to its receptor. That opens the channel. The sodium flows in and calcium flows out. This is actually not what triggers the action potential. That only affects the membrane potential in that very small area around the neurotransmitter. But if enough neurotransmitters bind to enough receptors, we get electrical changes such that the sodium voltage gated channel open, and that's shown in the third figure from the left. So with these channels open, the sodium rushes in. We see at the graph at the bottom that the membrane becomes more positively charged, and this phenomena, the depolarization, then triggers the opening of the voltage gated potassium cells, and that's in the final pattern. Once those potassium voltage gated channels are open, potassium flows out, and this causes the cell to repolarize. And indeed, it will hyperpolarize. As you will notice there, the graph descends below the original line, and it is, if the cell remains hyperpolarized, it will be refractory to firing again. And in a moment, we'll see that the way neurons regulate this hyperpolarization is key to their neuronal firing rate. Now, of course, when the action potential propagates down the axon, as you can see in A, when it reaches the end of the axon, then it will open the voltage-gated calcium channels. These will trigger the action of a number of enzymes that will cause the vesicles containing the neurotransmitters to fuse to the membrane in the synapse and release the neurotransmitter. This is all discussed in the book. Now, that's how the action potential is created and fired. Let's now look at the different second messenger systems. And the first messenger system we're going to look at is one of the G protein systems. The principal G protein is a trimer. As you can see in the upper left, the alpha, beta, and gamma proteins are linked together, and the alpha unit is bound to a molecule of GDP. And when the receptor binds, when the neurotransmitter binds to the receptor, as shown on the upper right, GTP will displace the GDP, and this will cause a dissociation of the alpha subunit and from the beta gamma subunit. Now, these subunits can have enzyme actions in and of themselves, but what's more important is that other enzymatic activities are triggered in the cytoplasm that affects neuronal activity. Now let's look at this in terms of the adenyl cyclase system. So we'll see two receptors on the neuron at the top. One is linked to a G protein that has an alpha sub S subunit, whereas the other is linked to the alpha sub I. And in this case, the S and I refers to the action on the adenyl cyclase enzyme. So let's take on the left, if that neurotransmitter binds to that receptor, the alpha subunit will activate adenyl cyclase. Adenyl cyclase will convert ATP into cyclic AMP, CAMP, And two molecules of cyclic AMP will bind to the regulatory subunit of protein kinase A, causing the regulatory subunit to break away. That unit, catalytic unit, will then move into the nucleus where it will phosphorylate a protein called CREB. This CREB will bind to the CREB responsive element, or CRE, and that will initiate the transcription of DNA. And of course, that will ultimately lead to the creation of various proteins that are needed for cell function, or to affect more long-term cellular neuronal activity. Now, the adenyl cyclase is one major system. The second major system is the PIP2, or phosphatidylinositol biphosphate system. Here you see that the alpha subunit of the G protein is an alpha sub Q unit. When GTP attaches to this, it will activate an enzyme called phospholipase C. Now, the phosphatidylinositol biphosphate, or PIP2, is a glycerol molecule that has two long fatty chains on two of its carbons, and those stick into the membrane. They're hydrophobic. The other carbon is attached to an inositol molecule, which is a sugar, has lots of hydroxyl groups on it, so it's hydrophilic and stays down in the cytoplasm. The phospholipase C will split the molecule, the PIP2 molecule, into the DAG molecule, which stays in the membrane. Of course, it's called diacylglycerol. That will activate protein kinase C, which then leads to various enzymes. The inositol triphosphate molecule, or IP3, then floats free in the cytoplasm and can interact with a receptor on the endoplasmic reticulum. This will change the activity of, this will change the level of intracellular calcium, and this will activate a number of enzymes. An important one is calmodulin, and again, numerous enzymes will be activated that will affect the nucleus and gene transcription, as well as various other neuronal functions. Now, neurotransmitters that work through the adenyl cyclase and phosphatidylinositol biphosphate system are often then referred to as neuromodulatory because they are not directly affecting the action potential. They are having more subtle effects that will affect neuronal function over the long term. Now, in the text, I'd like you to follow along carefully with this particular figure because it sort of shows us the role of the two of the neuromodulatory neurotransmitters, norepinephrine and acetylcholine. So in the graph on the upper left, let's imagine a neuron in a dish. We put some glutamate into that dish. Glutamate will bind to an ionotropic receptor and initiate a action potential. If we then put norepinephrine or acetylcholine in the dish, we won't see any particular activity right away. But now if we inject glutamate, we'll see that we get a much stronger train of action potentials that are fired. How does this come about? We'll now look at the lower part of the figure. You can see that norepinephrine binds to its neurotransmitter. It activates adenyl cyclase. ATP is converted to CMP. CMP activates protein kinase A, and in addition to potentially going to the nucleus and affecting gene transcription, it can actually phosphorylate one of the voltage-gated calcium channel and make it less permeable. Well, if less potassium leaves the cell at the end of the action potential, then the cell will not hyperpolarize as much and will be ready to fire again. Now, how does acetylcholine achieve this? Well, it binds to a receptor that's linked to phospholipase C. It cleaves PIP2 into IP3. and it then activates protein kinase C, which actually does the same thing. It will phosphorylate the K channel and make it less permeable. So here we see two different neurotransmitters achieving the same effect through different mechanisms. And also bear in mind, it is the type of receptor and the second messenger system it is linked to that determines the ultimate effect. Both norepinephrine and acetylcholine will bind to other subtypes of receptors that ultimately will have different effects. So it's not the chemical of the neurotransmitter itself that decides what it does to the neuron. It's the type of receptor and the type of second messenger system that's involved. Now let me blow your mind a little bit with the TRK system, since you are probably not that familiar with this system. Here we have these sort of protein sticks, as it were. That's a number one, that pierce or go through the membrane. And close to it is an enzyme called RAS. I won't get into why it's called that. It is bound to GDP and is thus inactive. When a growth factor binds to the TRK receptor, as you see in 2, GTP displaces GDP, and that causes 4 phosphate groups to bind to the TRK sticks. This triggers a whole array of complex interactions. One is that phospholipase C can be activated, and we can get the changes in calcium that I discussed in the prior slide. But on the other hand, the phosphorylated TRK system can attract all these various so-called scaffolding proteins. These are things such as SHC, IRS1/2, having nothing to do with the Internal Revenue Service, of course, GRB1 and 2, and SOS, so-called son of sevenless, so named because it affected the seventh eye in fruit flies. But I digress. Ultimately, all of this will affect an enzyme called PI3K, which now will go to that PIP2 molecule, and rather than splitting it too, it will phosphorylate it and create PIP3. This will lead to the activation of AKT, which will trigger a massive enzymatic cascade that will lead to transcriptions. And I have a little video that kind of describes that in a more 3D sort of look.
Aberrant activation of the phosphatidylinositol 3 kinase pathway, or PI3K pathway, has been widely implicated in many cancers. And increased activity of this pathway is often implicated in resistance to cancer therapies. In cancer cells, uncontrolled signaling through PI3K can occur by multiple mechanisms, such as mutations, amplification of tyrosine kinases, or of PI3K itself. PI3K signaling is initiated by the growth factor binding to the tyrosine kinase receptor, leading to receptor dimerization. As a consequence, lipid kinase PI3K is recruited to the internal docking site and becomes activated. PI3K next converts membrane lipid PIP2 to its active PIP3 form, which in turn leads to activation of a key signaling kinase, AKT. AKT promotes cell growth through protein synthesis driven by mTOR signaling and reduces cell death by blocking FOXO activity. Genentech Bio-Oncology is actively researching ways to inhibit PI3K signaling.
Now, you may say, well, why are we interested in an oncological process? Well, the reason for that is that this AKT can have influence on the so-called cell death or apoptosis enzymes. And neurons are constantly dying, or many cells are dying and reborn. Uh, if these are unduly activated in neurons, we can get cell death. And there's growing evidence that conditions such as PTSD or severe mood disorders are associated with cell loss in the hippocampus in particular. So interrupting this process here might be very important in treating psychiatric disorders that arise from a chronic and aberrant activation of the stress process. Now, we already know that mTOR might be involved in certain forms of autism. Uh, Remember, or that autism is one disorder that in a substantial number of people is associated with excessive brain size. So perhaps there is excessive neurogenesis that could be relevant, particularly in the neurodevelopmental disorders. All right, now let's talk about the steroid receptors and the neurosteroids. As I mentioned earlier, perhaps the most famous neurosteroid is cortisol. When cortisol enters the cell, it binds to the glucocorticoid receptor, knocks the heat shock protein off that receptor, and then it can enter the cell where it will trigger the transcription of many different proteins. This process, when you are in an acute stress situation, will increase glucogenesis. That makes sense because you probably need more glucose to meet that situation, but also will temporarily decrease or shut off your inflammatory response. Now this is a mystery. If stress becomes chronic and cortisol is being activated for an extended period of time, this may explain why chronic stress tends to be associated with a number of chronic diseases. We don't quite have all the pieces together yet, but we're going to look into this in some detail in subsequent lessons. We're particularly going to be interested in looking this in terms of PTSD, because it is apparent that people have different variants of the glucocorticoid receptor which modulate their stress response and their potential risk for PTSD. Now, we also need to start paying more attention to the so-called neurosteroids. Clinically, the most important neurosteroid is Zulresso, or brexanolone, the new drug that is approved by the FDA for the treatment of severe post-pregnancy, postpartum depression, excuse me. The neurosteroids are broken down into three major groups: the pregnanes, the androstanes, and the sulfonated. We'll worry about the chemical distinctions later. As you can see, the brelexone and allopregnanolone, those are the same thing. They're chemically identically. And in pregnancy, this neurosteroid rises both in plasma and in brain, drops very quickly. It may well be that certain individuals, that drop in allopregnanolone is what predisposes them to postpartum depression. I want to talk about this disorder more now that we are starting to understand the neurochemistry of it better. I will pose this question to you. This seems to be, from an evolutionary perspective, a very bad thing. Why would mothers get depressed right when their newborn arrives and the newborn is in the greatest state of need? It would seem to be very counterintuitive from a natural selection perspective. Food for thought, and we'll come back to that. Now, the problem with steroid chemistry is it brings you nightmares from organic and biochemistry. Let's not worry about these chemicals on the top, other than that this is allopregnanolone or brexanolone here. This is its full chemical name, 3 alpha 5 alpha THDOC. The intriguing part to keep, or the important part to keep in mind, is that uh, these neurosteroids can come from the periphery, but also the brain, the neurons produce these neurosteroids themselves, and they're present in both males and females. This here gives you the legend for the previous slide. It might be intriguing to find that there are benzodiazepine receptors on the mitochondria of the neurons, so they're not just on the GABA neurons. Now, what exactly does allopregnanolone do from a chemical perspective? So again, here you see the neurosteroid here. When it is released from the neuron, from the GABA neurons, it modulates the GABA receptors and enhances their effect, such that you potentiate the GABA, or the inhibitory effect, in GABA neurons. Again, this is the legend for the previous figure. I'll be sending you the PowerPoint and you can read that as your leisure. Now, we are interested in this particular area, and I did review this for the Prite, but alpregnolone is a GABA A positive receptor, now approved for the treatment of postpartum depression. But all of these agents here are also either GABA A positive or negative modulators, and they have a number of effects. They can be sedative, they can be neuroprotective or anticonvulsant. Pregnenolone sulfite has been speculated to be a memory enhancer. Now, all of these things have been looked at the level of animal studies. We don't know where they are in the pipeline as far as the treatment of human disorders, but certainly it's possible that allopregnanolone or brexanolone is sort of the first of many treatments to come. And that can be seen here, where in depression, in both males and females, you get decreased alpha 5 alpha A reductase expression in postmortem brain. That's the enzyme that produces allopregnanolone, and you get decreased levels of allopregnanolone in CSF and serum. Similar phenomena with allopregnanolone has been noticed in PTSD. And right now, acutely, for brexanolone, the response rate is much better in postpartum depression. Also a quicker onset. So that concludes our overview of the major methods of neuronal communication. This will now, in subsequent lectures, get into the individual neurotransmitters.